Okay. I think we should get started. I'm delighted to welcome Dr. Scott von Epps to this MIDAS COVID-19 seminar series. Scott is an assistant professor of emergency medicine, of biomedical engineering, of macromolecular science and engineering. He's an associate director of MCERC. He is, I believe, an associate director of the Biointerfaces Institute. And he has been, he's an expert on cardiovascular medicine and modeling, and has been studying these issues in the trenches as in the context of COVID-19. And we're eagerly looking forward to hearing what you have to say, Scott.
Excellent, thank you. Well, I will, let's see, make sure my slides advance here. There we go. So I will start with the obligatory disclosure slide. With what I'm talking about today, I don't have any specific disclosures related to conflicts of interest. So the outline that I'm going to go through, the big first one here is like, who am I? Who is this guy that's talking to you? And you know, what am I bringing to the table today? And then I'll go through sort of the motivation of why we're here in this coronavirus and what that means and sort of my take on that. It then sort of leads into sort of two major things or ideas that I think of when it comes to machine learning in healthcare. And one of them is this idea that there's a huge promise of an enormous amount of data in the healthcare industry. The problem with it being that we have lots of data, but there are very ill-defined labels and the context is quite difficult to obtain, making it difficult to actually sort of get any major traction in machine learning in healthcare when it has been much more successful in other industries. And then I'll go through a couple of examples of things that I have been involved in that sort of incorporate machine learning with infectious diseases and those types of things. Going forward. So who am I? Well, when I was a kid, this movie came out and I wanted to go to space camp. I sadly never made it, but I still sort of always dreamed of working for NASA or JPL or something like that. And then when The Martian came out, movie, it only reinforced that. I keep telling my department chair that if NASA gives me a good enough deal, I may head and work for them instead. We'll see, still may be possible. But sometime during high school, I found something that was a little bit cooler than rocket scientist that I wanted to study. And that came from an anatomy and physiology class in my junior year in high school, where we did a complete dissection of a cat, and which you can see here. And that just amazed me. I found that to be so fascinating, and sort of then decided that this was an area I really wanted to pursue, perhaps in biology or medicine. So I went to college at the University of Pittsburgh and I was a student there for 13 consecutive years, actually. I managed to get undergraduate degrees in molecular biology and chemical engineering. Back then, biomedical engineering wasn't a thing as an undergraduate, became a thing while I was there, but did not exist when I started. Then moved on and started the MSTP program there, where I got my PhD in bioengineering. And my focus there was on coronary artery dynamics and atherogenesis. And I did a sort of very detailed computational and experimental study for that. So I am at least got a little bit of computational modeling chops to hang out with you guys. And while I was there, I spent a little bit of time in the pool swimming for the University of Pittsburgh. So during that time, I also got married. And after finishing all of that, my wife said, "So are you going to get a job yet?" And well, sort of. I did start working for these 3 hospital systems, but only as a resident in emergency medicine, which is probably quite similar to being a graduate student all over again. Similar hard work and long hours, and you're really a trainee. And while you do receive a stipend, you're working pretty darn hard. It's hard to call it a job. You're much more in training. But I got to do some fun things there, like fly in the helicopter, which you see there. And to give you an idea of how old this is, this little guy that's in my arms there is now getting ready to start middle school. And the helicopter in my background, if you're looking at the video of me, is a totally new helicopter, different than the one that's in this one here. I also did a fellowship after completing my residency where we focused on developing specific engineered surfaces to prevent medical device infection. And so I have this sort of long background of sort of combining engineering and medicine, and started off in sort of the cardiovascular space, and have sort of migrated more toward infectious diseases. And as an emergency physician, I'm particularly interested in those that are life-threatening. And so as a good academic, I am collecting my affiliations. So I work in emergency medicine. I'm part of the Biointerfaces Institute, part of the leadership of MCIRC, biomedical engineering, and macro. So I'm doing my good job of filling out my CV appropriately. But to be clear, I want to say what I am not. So, because I'm going to mention a lot of things and describe a lot of stuff today, but I don't want it to be construed as that I am an expert in these things. And in particular, I'm not an infectious disease expert. I'm an emergency physician, and I see lots of infectious diseases, and it is the focus or motivation for much of my research, but it's certainly not my medical specialty or the epidemiology of it as we get into talking a little bit about coronavirus. I'm also not an expert in bioinformatics or machine learning or data science. What I am, and this is sort of the classic description of an emergency physician, is that I'm a jack of all trades, and I suppose you could say a master of none. But in this particular case, and in what I do, and hopefully what I'll try to convey to you, is the need for somebody to live at the interface, which is why biointerfaces is such a good place for me to be, both literally and figuratively, in that having a background in engineering that had a fair amount of computational modeling as well as biological experimentation. It included animal modeling, and then my clinical background. I really sort of can speak the languages back and forth, and I hope that you will find that I am at least versed enough to sort of bounce back and forth between the areas of data science and infectious diseases and clinical medicine and basic research as well. And I hope that one of my jobs is, even if it is not my primary research goal, but to provide interface in between various collaborators of very disparate research specialties. So why we are here, how we got started on this, is this idea, our motivation is related to the coronavirus. I'm sure lots of you have seen this picture here. The current disease known as COVID-19 is caused by the SARS-CoV-2 virus. On December 31st of 2019, China actually reported this for the first time to the World Health Organization as an unknown cause of pneumonia. This is the report that you see there in that picture of Wuhan from January 5th describing that. So less than 6 months ago is when this was sort of unknown and we were still sorting these questions out. As of yesterday, when I looked these numbers up, since that time, there are over 9 million cases and almost 500,000 deaths worldwide. Here in the United States, we have almost 2.5 million cases and 123,000 deaths. And that certainly went up again overnight. This is the graph that keeps me up at night. You know, as an emergency physician, we saw these patients. At one time, the University of Michigan health system had 230 patients in the hospital with positive coronavirus. And so we were really taking care of these people. And this trend here for the United States is quite troubling to me. We are, while we are opening up and things are looking good here in Michigan, we are far from out of the woods. And so I keep this in mind as we go forward. But the thing that I find that the coronavirus has brought into the most stark reality is the difference between how fast this virus, or any virus for that matter, can spread versus how quickly we make scientific discoveries and innovation. I don't know how many are familiar with Dr. Markle, who's actually faculty here at Michigan. He actually made this quote about how germs have always traveled. We've had lots of pandemics, but now that they are traveling at the speed of a jet plane, he actually made this quote in 2014 related to the Ebola virus pandemic or epidemic that was going on at that time. And it's quite prescient now as well. The problem with this is our fear of this massive spread has led to us to some perhaps rushing of scientific discoveries and things like that. In general, what I worry is that we're doing a lot of undisciplined interpretation of limited data. And for that reason, we see things like this, which is the hydroxychloroquine. It's been in the news. People have either hate it or love it. And I won't get into the politics of that. But clearly, there were two very high-profile studies, one in the Lancet, one in the New England Journal. So these are very high-impact journals, and both of them were retracted. And so our sort of human fear instincts to try to move forward with what the exact results of things are have led us to sort of probably moving faster on scientific discovery than we necessarily should. And I think this is of particular concern. Now, there are probably ways we can do it smarter and more efficiently so that we can get answers and control the spread faster. But this stark difference between these two speeds, I think is the thing that I continually think very hard about. So this speed towards scientific discovery and innovation was quite evident here in our own university, University of Michigan. So what I've done here was I actually copied the titles of every single COVID-related research project that was put forth on the Office of Research webpage. There are actually 316 projects listed there. And that is literally just the titles of all of them in very low point font, 6 pages worth. This is quite outstanding. You know, I'm guessing that none of these people, or the vast majority of them, were doing any type of viral research, including myself at the time, who focused more on bacterial side of things. And I do have, and I think it's somewhere on this page, somewhere down around here, one project of my own looking at antiviral coatings for surfaces to control the spread related to high contact surfaces, which I won't discuss in detail today. But as we are just sort of getting it off the ground. But this is the sort of what I'm bringing at is we can certainly initiate and do amazing research and move things forward. But I think we have to make sure that we maintain the rigor that we have always done in moving forward with science. Of course, it's important to keep coronavirus in perspective with sort of prior pandemic research or prior pandemics that have happened. This particular plot that is shown here is actually back from early in May from the World Economics Forum. And at that time, I think it was no, I'm sorry, this was actually back in March, I think, very early. There was only 4,700 deaths from coronavirus at the time. So clearly we have moved well beyond that. So if we take this spot here and we move it to where it should be, it's still quite small, somewhere right around here at about 500,000 deaths between the swine flu and the 18th century Great Plagues. We have a long way to go compared to things like HIV or bubonic plague or smallpox, which are in the tens to hundreds of millions of deaths associated with them. But we're certainly not out of the woods at all from that. But what this sort of gives an idea is this is not a new problem, and it's been going on for quite a while. The thing that irks me the most about these particular ones is that the last 3 are all created by coronaviruses. The current COVID-19, the Middle Eastern respiratory virus, and the severe acute respiratory syndrome virus, the SARS virus. All of them are coronaviruses, which is certainly something to think about going forward. But there are other emerging threats that we have to think about, and I think the only way to really tackle these problems is to be on top of them ahead of time and sort of build infrastructure toward sorting them out. One of the major emerging infectious threats that will always be there for us is antibiotic resistance. And so this picture here is of Alexander Fleming. He discovered penicillin back in 1928. And since then, we've had this sense that if it's a bacterial infection, we can cure it with antibiotics. Unfortunately, bacteria are quick to develop resistance, and during my lifetime, there's been significant increase of resistance and a significant decrease of brand new antibiotics. In fact, really during my lifetime, there's only been 1 or 2 new classes of antibiotics. All of the other new ones are slight modifications of the classes we already have. And if it's a slight modification for us, it's a slight modification for resistance for the bacteria. The other thing that is probably an emerging threat that gets less press is this idea of medical device infection. So we are putting more and more pieces of plastic and artificial materials into patients to prolong their lives, and they're doing amazing work between pacemakers and artificial valves and prosthetic joints, dialysis catheters. These things are saving lives, but they introduce a particular nidus for infection. Here we have a central line. This is an electron microscope image of a biofilm of bacteria growing on a ventricular peritoneal shunt. And the problem here is with biofilms. Biofilms that grow on these various devices, such as the catheter here on the far right in a human, or the catheter on the rabbit model done in our lab, they provide significant protection from the regular immune host defense. They also prevent antibiotics, which would normally work. This is not resistance, it's a different level of protection by restricting the diffusion of the antibiotic, reducing the metabolic activity, or actually degrading these small molecules. And the other thing that biofilms have are these things called persister cells, which I liken to the stem cell of a biofilm, which if you eradicate most of the biofilm with the exception of a few persister cells, they can repopulate the entire film. So with all of that in mind, I think it's important to now sort of move on to some of the things that we maybe can think about in terms of healthcare and how we might go on with big data. The clear message that I take away from this, and it may be unique to me, and others can always differ with me on this, is that we have to think about problems like emerging threats that can spread very quickly if we do not have a treatment for them, like coronavirus. We have to think about them now, and we have to be moving forward with plans as if they are here. If we wait until we already have infections and people are already dying, we are way behind the 8 ball on that one. So let's talk about the promise of big data. In clinical medicine, there is certainly a lot of data. There is no doubt about that. So this picture is from a patient in an intensive care unit, not unlike the one that we have in the emergency department, where there is an enormous amount of information between information from the monitor, the EEG, the EKG, the ventilator, labs, imaging, and then the actual physical exam related to the patient. So, it's been estimated that up to 200 time-related variables are occurring. The problem is any given physician really can only think about a relationship with 2 variables at a time without any other kind of assistance. And this is exactly sort of the idea behind bringing data science and machine learning to this. The amount of data sort of spans many different scales. So for instance, at the very smallest level, you can imagine a molecular dynamics simulation here of just, this isn't even thousands of atoms, this is just a few hundred of a small peptide toxin released from Staphylococcus aureus that is moving in 3 dimensions. But there's an enormous amount of data in that one little simulation right there. And this gets exponentially bigger when you start adding the various things that this particular toxin binds to or aggregates with itself to form fibrils. Moving up the scale, the central dogma of biology is clear here that, the human has about 25,000 genes, which then sort of, in theory, translates to a single protein. But when you start to include variable splicing, post-translation modifications, various other sort of modifications and polymorphisms, that could sort of get on the order of about 1 million different protein species in a given cell along that range. Still moving up the thing, there are 200 different types of cells and about 3 to the times 10 to the 13th total cells in the human body, each one that are sort of working along the range of that central dogma and at different scales. And with the advent of sort of single cell sequencing, you can get at the single cell level, but understanding that that is sort of representative, a tiny representative of a much larger population and what that means. Added to this, which is probably less well described, is the human microbiome, which to some degree is both an advantage and also unfortunate that we don't know more, since it does actually, there are 10 times as many bacterial cells in the human body as there are human cells, and significantly more genes at about 10 million genes. And then finally, right, when you sort of expand this out to the next sort of level, we get to the population, of 7 billion people in over 195 countries of different cultural, ethnic, racial backgrounds, as well as experiences that sort of contribute to these things. And then the electronic health record. I can tell you from experience, I spend an enormous amount of time interfacing with this thing, putting data into it, reading data out of it. It is certainly a lot of data, but it is, to be clear, it was not meant for us to do research, or at least that's not how it was designed. It was not designed for simple data abstraction. It was actually not even really well designed for good medical care to help the physician. In reality, most of what it does is help us with billing, which is quite sad commentary on it. And while we can utilize it, the fact that it wasn't specifically designed for the questions we want to ask from a scientific standpoint leads to many difficulties in sort of how we think about this problem moving forward. So next I want to talk about the peril of ill-defined labels here. And this is really an idea related around data is valuable, but labels and context are really truly priceless. And that's a quote that I sort of stole from Kevin Ward here. And the example I'm going to use for this is sepsis, because this is a disease that is been in the news. It's very exciting. People are working very hard on it, but we've not made any real game-changing things, any game-changing technologies or management related to sepsis, really since I've been practicing medicine, and probably since the early 2000s. Interestingly, many patients with coronavirus are dying of respiratory failure, but ultimately, it is a broad immune response that is causing this thing, and viral sepsis is really sort of what we're seeing in these patients. So it has a lot of relation to the current epidemic or pandemic that we're seeing here. So what is sepsis? Well, it's certainly a problem. There's 19 million worldwide cases, 1 million in the US, 250,000 deaths per year. It is the #1 cost, 20.3 billion, and these are actually old. They're probably much higher now. So it is a big problem. And if you survive, there's a lot of ongoing problems. 3/4 of people have long-term issues related to their prior sepsis. There's a high risk of recurrence. There's a lot of PTSD related to being in the ICU and on a ventilator and under the medications and the delirium that had prior caused. And there's really little in terms of support for these folks, right? There are cancer survivor groups and heart survivor groups, but there really isn't for sepsis, which is troubling given that it is probably a bigger problem by numbers. But what is sepsis? And this is sort of the real question, and the reason why it's very hard to study from the standpoint of big data and machine learning for predictions, because you got to know what your categorizations are ahead of time. You got to define the labels, and that's quite hard in sepsis. So the original sort of definition of sepsis was this systemic inflammatory response to infection. And it was defined by these vital sign changes related to your fever or your heart rate, your breathing rate, or whether or not you had a low or high white blood cell count. And then if that was associated with a known infection, we called it sepsis. And if you had any organs that weren't working like they should, we called that severe sepsis. And then if it also caused your blood pressure to be low, then we called it septic shock. And then more recently, the sepsis 3 guidelines came out and changed this definition. And I won't get into the details as to why that is. But the process of that is that starting with a known infection and then having this SOFA score of change of greater than 2. And the SOFA score is defined by this table that I added there. And that was defined as sepsis. And if you had a low blood pressure requiring vasopressors, or you had a high serum lactate, we called that septic shock. But getting this SOFA score can be difficult. And so they made this simpler one called the Q-SOFA, which was a little bit easier, but it predicts something a little bit different. But the difference between these two definitions is really one of diagnosis versus prognosis. And so when they're compared, it turns out that the original systemic inflammatory response is very good at diagnosis. It's actually quite sensitive, but not very specific when it comes to diagnosis. However, the Q-SOFA was much better at prognosis. That is, it probably missed folks who had it, but when you did have it, it predicted a poor outcome. And in this particular case, that's usually mortality. So it really depends on what you're trying to decide. And this is one of those things about sepsis that I think is probably more troubling, is that as more and more people try to predict whether you have it or not, that they lean on these particular scoring systems without regard for what it is that they were really designed to do. So in the treatment of sepsis, resources matter, but we don't know which ones. The probably most revolutionary study related to sepsis was this one in 2001 by Manny Rivers, done right down the road there at Henry Ford, excuse me, which demonstrated that if you applied this enormous amount of resources, this early goal-directed therapy, you could drop the mortality rate of sepsis and severe sepsis and septic shock from 46 to 30%, which is huge. In 2015, they basically sort of repeated the trial where they used this early goal-directed sort of protocol and then just compared it to what we were doing. And there really wasn't any change. The reason for that is because a lot of what was going on in the early goal-directed therapy in Manny Rivers had become common practice. And so people were doing more titrating of the therapy. And it turns out if you do everything that the early goal-directed therapy says to do, you're probably spending more money and doing more harm for patients without any mortality benefit. And so it turns out we don't know what the exact sort of specific things that we needed to do for these patients were. If you do everything, you can certainly help, but there's probably significant diminishing returns and potentially harm that come from that. So I want to tell you a little bit about my world and how I think about sepsis, because in the emergency department, we work from very limited data and a lot of unknowns, and have to make a lot of decisions on that sort of sparse data that is coming at us in real time. So the scenario is that a typical septic patient comes in an emergency department. They may be febrile, they have a fever, their heart rate's high, their blood pressure might be a little low. They may be confused and can't tell you, what the exact sort of onset of their symptoms were. Many times I won't know the source. Is it in their lungs? Is it their skin? Are they have a urinary tract infection? Do they have meningitis? I certainly won't know what the bacteria is, and I definitely won't know which antibiotic to use specific to that bacteria at the time they come in. But the goals, according to CMS and the Surviving Sepsis campaigns, and now the Sepsis 3 thing, are to measure lactate. We're going to give antibiotics to basically cover everything, obtain blood cultures to look for the bacteria, give IV fluids, and we should do all of this in the first 3 hours. And then if they still are struggling and their blood pressure is low, give vasopressors within 5 hours and repeat the lactate to make sure that things are getting better within 6. The problem is I don't know which resources to deploy at this point to know if somebody's getting better. When somebody's super sick, it's easy. I know that they're really sick and I need to work very hard, but many patients look like they might be sick or have an infection but don't actually meet full criteria for all of these things. And I don't want to do more than is absolutely necessary based on the sort of prior studies. And so do I put a blood pressure line in this person? Do I put a large bore IV in their neck? Do they need to go to an intensive care unit, or can they go to a moderate or a general care floor? How much fluid do I give them? How much nursing care do I need to give these people? These are very difficult questions. And the thing that is sort of concerning with all of this is that the Centers for Medicare and Medicaid basically tell us that we have to do all of these things, essentially in order to get paid, or to be on the good status, which affects our reimbursement. Interestingly, we've done this kind of thing before. We did this for pneumonia many times, and unfortunately, they forced a sort of 4-hour rule that is, you had to give antibiotics within 4 hours of their presentation. This was ultimately relaxed because we found out that we were probably overdiagnosing it to make sure we didn't miss any, and we were probably using antibiotics on a lot of patients that didn't need it. And it ultimately was probably leading to harm, and this went away. I fully suspect that the CMS rules about sepsis will be the same way. That because of this, we're looking for sepsis. We're finding it more often, at least as best we can, based on an unclear definition. But we are treating it more frequently, and therefore we may be causing harm. The problem is sepsis is a bit like, as Potter Stewart, the Supreme Court Justice, said about pornography, right? I can't sort of fully describe it. It's not clear to me. I can't put a sort of thing on it. I can't embrace a specific description, but I know it when I see it. And that is the difficult part in applying a very a disease that can certainly benefit from a deeper understanding of the multitude of data coming in on it, but without me being able to provide you with specific labels and categories for whether or not a patient was actually septic or not, it's quite difficult to pull from the medical record. The other issue that I think comes in when I'm talking about the perils of these is just the simple act of extracting really decent data from the medical record is quite hard, and it seems like it shouldn't be. For instance, what is the length of stay of a patient? Well, when a patient comes in, there are many time points associated with them, and how you specifically use them are difficult. So for instance, is it the time that they walk in the door to the emergency department? Is it the time that I get them admitted? Is it the time that I get them a bed? When they get discharged, is it the time that the doctor signs the order? Is it the time that the nurse reads the instructions to the patient? Is it the time the patient actually leaves? And these are not clearly defined and sometimes not available from the medical record, depending on how the record is designed. Similarly, ICU length of stay is a very important sort of data point that we like to know from a research standpoint. But it's hard to know physically where a patient is at any given time during their stay in retrospect when you abstract that from the electronic medical record. Ventilator days. So how many days is a patient on a ventilator? This is not easily abstracted just from simple pieces of information. It's getting better, but a lot of times you have to actually go into the notes and find out when a patient was intubated and when they were extubated, which might come from actual physician adjudication. Vasopressor use is another one. Many times patients are on these medications intermittently for a couple of hours and then off again, or they're on for several days at a time continuously. And sorting out their sort of sort of area under the curve of vasopressor exposure in terms of dose and time, it can be quite difficult from the record. Similarly to antibiotic exposure. The other thing is extreme events are quite rare, things like cardiac arrest. And so if you had a prediction model where you just said that nobody ever had a decompensation or cardiac arrest, you'd be right a lot. And so thinking differently than sort of the typical area under the curve kind of measures are necessary for these extreme events, but are quite important on sort of a human level. And the other problem is artifacts are common. I'm constantly irritated when a vital sign in the chart doesn't make any physical sense. I've looked at the patient, there's no way they can be breathing 80 times a minute. But somebody has made a typo and hasn't gone back and corrected that. And so when you abstract that data from the electronic medical record, you would never know the context to know if that was true or not. All right, so in the last little bit here, I'm going to talk about a few examples of where we've been taking infectious diseases and sort of applying some machine learning and data science related to them going forward. So the first one is on the pathogenicity of blood isolates of Staph epi. So Staphylococcus epidermidis, it's sort of a cousin to Staph aureus, which more people are familiar of. And the question is it a good guy or a bad guy? Well, it's actually the most frequently isolated species from sort of human epithelia, right? That's your skin and your eyes and the inside of your mouth and the inside of your nose. It's just there, you're living with it. And it's probably not hurting you. In fact, it's probably helping you to some degree. The problem is it's also one of the most common causative agents when it comes to indwelling medical devices because it's so good at building biofilms. So because it lives on the skin, but it can also cause these infections, which ultimately lead to bloodstream infections. When we find it in a blood culture, it's very hard to know whether or not it's a pathogen or just a contaminant from them putting the needle in your arm to measure the blood culture in the first place. So we were able to develop a library of isolates that sort of fall into these three categories. One is commensal strains that we got from healthy people who put their thumbprint on a agar plate. We also obtained pathogenic strains, and these came from people who we thought really did have disease. They had two sets of blood cultures that were obtained at the same time. The patients were symptomatic and by CDC criteria were considered true positives. And then we have contaminants where maybe this patient had symptoms, it wasn't clear to us. It was enough that we decided to get a blood culture, but it only showed up in one of the two bottles, which wasn't enough to call it a full positive. And so we consider these false positives or contaminants. So taking this library, my prior mentor had actually looked at a technique called multi-locus sequence typing that sequences or partially sequences 7 housekeeping genes. And then assigns them a sequence, and from that you can then give an overall sequence to the specific isolate. This is sort of a 7-dimensional categorical measurement, and using this, we then applied some machine learning algorithms, very simple things, and like support vector machine and naive nearest neighbor, and was able to sort of generate this denogram there, but the diagnostic accuracy was only 73%, and part of that is because of the number of different types of machine learning, or I'm sorry, the number of types of sequences that are available that might only be there in 1 or 2 isolates. The problem with this is that this has really nothing to do with whether or not it's infectious. You're focusing on only the housekeeping genes, and so we asked, what about specific phenotypic measures? And I'm going to go through these sort of very fast and sort of get to the meat of this. One of them is do you have antibiotic resistance? We think that pathogens are more likely to have antibiotic resistance than commensal strains or contaminants. What about your ability to form a biofilm? This is sort of the superpower of Staphylococci, is building a biofilm. The other one is how well do you grow in human plasma? If you are sort of better suited to grow in plasma, you're more likely to be a blood pathogen rather than a skin contaminant. And then the other thing is if we're going to focus on genes, we should at least focus on specific virulence genes like methicillin resistance or those genes which sort of aid in binding to human tissues. So the question is, are you a pathogen or not? These were the groups here. And we think of contaminants and commensals as being really the same population. It's just that the commensal strain sort of got moved into a blood culture model during the process. And so using a analysis of similarity study, we were able to demonstrate that the difference between each of these groups was actually more than the difference within the groups, which is at least helpful, and that the pathogens were actually sort of this more or less diverse group than either the commensals or contaminants. And so we felt comfortable sort of putting the commensal and contaminant groups in the same bucket, at least for doing a binary sort of study. And so using all of those different variables of phenotypic studies, such as the growth curves and the biofilm forming capacity and antibiotic resistance, and we did sort of built several models using binomial logistic regression, support vector machine, recursive partitioning, nothing that was very high tech, but just sort of applying them. And so the addition of these phenotypic tests to the multi-locus sequence typing certainly improved our accuracy in the so approaching sort of 90%, which is significantly better than we were doing before. More importantly, what we found is that oxacillin resistance, which is actually how we measure methicillin resistance when you've heard of MRSA or methicillin resistant Staph aureus, was the sort of most important variable, and it showed up in all 5 of the models and sort of outperformed the sequence type that we saw more. And in general, the antibiotic resistance, which is something we measure anyway in all of these bugs, was quite important in doing that. All right. Second one is in this idea of predicting protein-protein interactions. So as I mentioned, there will be a never-ending need for antibiotics. As soon as we make more antibiotics, bacteria will evolve, things will change. And so as we sort this out, we're always going to need to be thinking about this. The problem is the pipeline for discovering new drugs gets narrow very, very quickly and takes a very, very long time. And as I was saying, the drug discovery, scientific discovery is way slower than the development of sort of infectious diseases and the spread of them. To make matters worse, and to try to get around this a little bit, is this idea of the use of nanoparticles or nanomedicines as antibiotics, which we have focused on extensively since I've been at the University of Michigan. However, nanoparticles add even more complexity to the problem, and trying to find ways to better understand how a nanoparticle might interact with a particular protein target that it is aimed for is important, so that we are not performing a trial and error system of screening of thousands of compounds to come down to one that takes 2 decades to get there. And so starting with this idea that we looked at zinc oxide nanoparticle shapes and how they interacted with a particular target, in this particular case, beta-galactosidase, we see that there is a shape dependence of the nanoparticle. So same material, zinc oxide, but different shapes created much different inhibition of the enzyme. And this shape dependence mirrored the antimicrobial properties, where we see growth curves of pyramids that almost completely inhibit growth, versus plates, which sort of have a dose-dependent growth, and spheres which had very little effect on the growth of the bacteria. So we're really trying to figure out ways there. What are the features of nanoparticles that predict a nanoparticle-protein interaction? However, there's not a lot of data about nanoparticles, and so we have to take a step back and work with something that we do know, which is the idea of protein-protein interaction. Most of the protein-protein interaction work in the machine learning environment has been based on the sequence, which we see here on the left. And that's just the primary structure of the protein itself, versus the actual 3-dimensional structure for it. And so we know that this particular amino acid that's here at the very beginning might actually be in very close contact to this amino acid over here. And that may be very relevant to the functionality. And we know that the shape of the protein is sort of key to its function anyway. And so in finding ways that protein-protein interacts is important. Secondly, if we want to translate this to nanoparticles, we need to think in terms of geometry, because nanoparticles do not have they are not a polymer chain of amino acids that can be thought of in sort of a sequence like this. And so working with a team as part of the Blue Sky program, which is a project that was led by Angela Violi in mechanical engineering, which is geared toward exactly this, accelerating the discovery of new nanomaterials as antibiotics. One of the things we did working with Al Hero in EECS was to convert a peptide shape into this sort of graph network shape, where you can actually bring into close contact or demonstrate the sort of proximity of particular amino acids based on their proximity to each other, and sort of preserve this shape and geometry in it. That can then be applied in this struct2 graph mutual attention network that they built, that was able to help predict whether or not two proteins and their shapes would then come together in an interaction. But here's where understanding labels that I mentioned, or context, is really important. So what exactly is a protein-protein interaction? It seems quite simple, as I tell you, it's two proteins coming together, and do they fit together? Do they interact? Do they do things? Except that when you actually look in these databases, the interaction type can be quite amorphous. It could just be an association, or a physical association, meaning they're near each other, they're on the same general area of the cell, or there could be a direct association, or a co-localization, or actual binding. And so in order to really understand that these things come together and physically interact, we had to focus specifically on the direct association and binding, and that required a lot of manual curation of the database. In addition, we had to understand the protein structure. And in the literature, there's many protein structures, and a lot of times proteins are cut into pieces in order to obtain their structure in the crystalline thing. So we actually had to go through and determine whether or not a protein structure was this versus this, which is actually the full, complete three-dimensional protein structure. This is only a portion of it. And so this is why sort of this idea that Kevin Ward has said multiple times, that data is valuable and context is priceless. Because it really did narrow the number of true positive protein-protein interactions going forward. The performance of this struct2 graph model was fantastic. It out-predicted many different models for protein-protein interactions in a balanced data set. It also outperformed in an unbalanced data set, which is really sort of the reality, where the vast majority of proteins won't interact with other proteins. There are very unique specific interactions, and so its performance in an unbalanced data set is probably more indicative of what we would be interested in. In addition, the prediction is more than just a binary yes or no, do they bind, or what is the probability of binding, but it was actually able to determine what the specific interacting portions of the structure were. And this is sort of a model of that, where we demonstrate a toll-like receptor molecule and its normal substrate versus a bacterial toxin which binds that receptor as well. And so a similar type pattern you could imagine for the coronavirus, which binds to a human angiotensin converting enzyme receptor through its spike protein, which is a sort of different substrate than that receptor's normal substrate. So this is the sort of strength of that particular work. All right, and I see I've probably gotten sort of long on time here, so I'm going to go through my last one real fast. And this is probably the sort of the beginning works, and I'm hoping to spend more time with this particular group, where I am very interested as a physician in anticipating when somebody is going to get worse because they have an infectious disease. As I said before, it's hard to predict how much resources should I contribute to a patient. I already showed you this slide here. This is sort of my world where I have to accomplish these things, but I need to know what resources I'm going to deploy to this patient. One of the most expensive decisions I make every single day is whether or not to keep somebody in the hospital and then which unit to put them in. And I have to make that decision very early on in the process. I'm expected to be right a lot, and the consequences are both huge on the financial end and even more drastic on the health end of things. And one of the major questions is, does this person need a lot of care such that they need to be in an intensive care unit versus moderate care versus a general care floor on the bed. And the major differences in there is how many interventions can you do for this patient, and what are the nursing ratios? Is there one nurse taking care of 2 patients, or is there one nurse taking care of 8 patients? For this, I look to Kayvon Najarian, who I have been working with recently, and a graduate student in his group, Olivia Algay, who we're actually looking at this not from infectious diseases, but this could be rapidly leveraged to them for monitoring patients in postoperative cardiac care. So these are patients who've come out of a surgery and they may decompensate. So these would be sort of quite analogous in terms of maybe not physiologically, but sort of predicting the same sort of decompensation event. And so you can imagine that within the electronic health record and the physiologic signals that I showed you earlier coming out of them, you can extract a lot of features from these waveforms from the EKG and the blood pressure monitor and the breathing monitor. One of the specialties of Kayvon's group is the use of this tensor-based dimensionality reduction to really reduce the feature space in working with it. And like I said, this is what I'm interested in, this idea of hemodynamic decompensation. And so these are this idea of these tensors, usually third or fourth or multi-dimensional higher order tensors for making these predictions. And so what they do here is they decompose this tensor in a way that allows them to sort of reduce the number of features here. You can imagine that in this particular case, if we look specifically at let's look at just an EKG for that instance. There are many features within that waveform. There's a time window, there are the features, and then they use a taut string to estimate the actual sort of signal or provide a approximation of that signal. And that can then be built into a third order tensor. And if you stack them along the fourth order tensor for each patient in the training set, you can get this sort of fourth order tensor. I will be honest, the math is sort of beyond me, as not a non-data set. I'm more interested in sort of what the results are. And looking at these, one of the things I see here is that utilizing this data, if you look, they are able to predict a decompensation event, which is sort of given by a sort of time 0, right? With accuracy area under the curve around 85% in their test data after their training, 12 hours early. I can do a lot with 12 hours. That's a lot of time for me to actually make an intervention. Certainly it's time for me to say, you know what, this patient's looking a little sicker. Let's put them in the intensive care unit, and maybe they don't actually decompensate in the end by making those things. And so these are the kinds of sort of benefits that you can imagine from that, and hopefully sort of applying similar type applications to the infectious disease population over the post-operative group will certainly have benefits similar to that. When it comes to higher order data, it's going to be important to work with them in a way that allows you to sort of preserve the information within them. And so if you're going to go into higher order tensor data, you know, many times for vectors, this was done by linear discriminant analysis. And so Kayvon's group has developed this Kempfness multilinear discriminant analysis methodology, which allows them to basically perform an LDA on the tensors. And where this was applied is on EKG data. And so if you take an EKG and you group the patient's EKGs by whether or not they're healthy or unhealthy, as in they've had a heart attack, or they have evidence of cardiomyopathy or heart failure, or they're in an abnormal rhythm or other obvious heart disease like pericarditis, can we use this sort of tensor data that we described on a higher order level to predict whether or not they fall into these classes? And indeed, this is the case, and they are able to do this with area under the curves of, say, 94%. Interestingly, and the thing that I'm quite interested to explore further is the idea that this is still quite powerful even with increasing signal to noise ratio. Having looked at a lot of EKGs and having patients that move around and a lot of artifact in the data obtained on these, I find this to be quite interesting. You know, the EKGs have a small computer in them that spits out a read on them, and they're almost always wrong. And that's why the physician reads them specifically. But if this is able to make this prediction even with this level of noise, I think is quite exciting, and hope to utilize this same technology sort of coupled with the other work for our infectious disease groups. So I think we are right at 2 o'clock. There are lots and lots of people to acknowledge. Several folks in my group, Matt and Usha, were involved in the zinc oxide work. Yichin has been involved in the molecular dynamics modeling and some other work in my lab. Shannon was the main driver of the work related to Staphylococcus epidermidis. I've already mentioned the work with Olivia and Kayvon, and of course the protein-protein interaction work, which is sort of currently under review at Bioinformatics, was led by Al Hero. That particular thing is part of the Blue Sky team, which is led by Angela. Particularly, I want to call out Mayank, who's the lead author on that thing. And Sumera and Shivani worked very closely with me in curating that database to provide the appropriate context. We've had funding from a number of sources, and I will take any questions. Thank you for giving me this opportunity to chat with you guys.
Perfect. Thank you so much, Scott. So we are at a time here, but I think we should definitely take a couple of the questions that have come through in the Q&A here. So if you don't mind, I can just read these off for you. So the first one we have is actually from Kayvan. He says, "Thank you, Scott, for the wonderful talk. You mentioned that one of the factors to consider for prediction using machine learning is the timing of ventilation, in particular for COVID-19 patients. Seems rather straightforward that machine learning on the patient data can predict, perhaps well ahead of time, who would need that ventilation. But do you think a similar approach could be used to predict when a patient who is using a ventilator would feel well enough to be removed from that mechanical ventilator?"
Yeah, I'm going to be honest, I think it's easier to predict when they can come off than when they can go on. And the reason for that is because we've already built all kinds of sort of scoring systems and measures for that the respiratory therapists perform, and they actually do it almost every morning, and they tell you how likely somebody is to come off. What I would say is we are very conservative about that, meaning we wait until we're absolutely positive that that person can come off the ventilator, and so because we're not very tolerant of reintubation. And I think we should probably push the envelope because we end up leaving patients on ventilators probably longer than they need to be. And finding that sweet spot is really the difficulty. It's where context comes in. Is you might be able to predict with X accuracy, but the comfort level of the physician sort of might drive that. And I think we need to sort of understand where we are on that. But we tend to be quite conservative, even in the setting of having some predictive tools.
Perfect. And then we'll take one last question here. We've got one from Dana King. So how feasible is it to apply modeling to real-time data in a clinical setting than to get that 8 to 12 hour window of time prior to predicted decompensation?
I think from a sort of physics, science feasibility, I think it's quite feasible. There are already systems in place running in the background, creating various scoring systems that run in real time on the electronic medical record. For instance, we have this thing called the Predict score, which gives you an idea of how sick you are in the emergency department. Just generally, are you really sick or are you not really sick? In the sort of 8 to 12 hour time frame, certainly if you wanted this to run in real time and give the physician that information, there are a lot of regulatory hurdles to, is this a diagnostic or is this clinical decision support? And depending on what that is and how much information and how much regulatory trials have to be done before you can deploy it as such, that's your hurdle, not the science or the physics of actually getting the information to the physician. And if, Dana, I hope that makes sense.
Perfect. Excellent, Scott. Well, I think just for the interest of time, we'll leave it there. I'll save these other questions for you, Scott, if you feel like reaching back out to the individuals that asked them just so they can get some extra clarification, and I'll turn it over to Jag.
I just want to thank you very much, Scott, for a wide-ranging and very informative talk. I'm sure that everybody enjoyed this as much as I did. Thanks again, and thanks everybody for listening in.
Thank you very much for having me. I enjoyed it, and I'm happy to chat with folks who are interested. They can contact me directly. I'm very happy to sort of, I'm a very collaborative guy, so please contact me if you would like.
Thank you all. Bye-bye.
Take care.